This video will cover how to solve some of the problems you may run into when re-annotating more difficult genes on your mitochondrial genome. Sometimes the most difficult genes to annotate are those found on the light strand or the negative strand of the mitochondrial genome. These require reverse and complementation of each nucleotide in order to find the correct sequence. In this example, I'll be using the NAD4L gene, which is a small protein-coding gene found on the negative strand. You can recognize that the file by Mitos has already given you the reverse and complemented nucleotides for genes which it recognizes are on the negative strand. The start location given by Mitos is where you will find the stop codon for your gene on the positive strand in the Mega software. As Mega only reports the positive strand, the location given by Mitos of the typical start is where you will begin to find the stop codon due to the reverse and complementation process. By going to the start location reported by Mitos, you can see the nucleotides in that location on the Mega file are complementary to those on the end of the sequence on the Mitos file. After pasting in the Mitos file sequence, you can see that both the start and stop need to be re-annotated. In order to find the stop codon, you will need to reverse and complement each nucleotide from Mega in this direction while adding to the end of the XPASI translate sequence. Paying attention to where the surrounding genes are located will help you determine how far you've gone into the next gene or how far away you are to see if you have any unusual gaps or overlaps. Adding a series of reverse and complemented nucleotides, I'm still looking for where the stop codon is. I will then try removing some of the nucleotides from the original sequence by Mitos to see if the stop codon is located within that sequence. Recognize this TAA as a typical stop codon, so I will now see if this is where the gene might possibly end and continue to pay attention of the location that I am at in Mega in order to properly report this stop location of this gene. Next is finding the start codon and start location for NAD4L. Go to the location of the second number in the Mitos file, in this case 8245, to find the region where I may encounter the start codon. This time I will reverse and complement the Mega nucleotides in this direction while adding to the front of the sequence in XPASI in a backwards direction. Just like with genes in the positive strand, your sequence should have both a start and stop codon. Although there are some exceptions to this rule, which are the genes that have or are presumed to have incomplete stop codons. In this case, you can refer to other literature in order to see if that's a common occurrence reported in other papers. You can also use this literature to compare your start and stop codon to see if those are typical for that gene. After comparing my results, I will put them into my finalized table, making sure to note that the gene is on the negative strand, the start and stop location, as well as the codons I found. These are some questions to go through and ask yourself if you happen to run into trouble while annotating your genes.